Today it's my pleasure to talk with a friend and someone who has common interest in brain aging, Alzheimer's disease, what's going wrong, and how an understanding of those problems may eventually lead to some novel approaches for the disease, either prevention or slowing the course anyway. So my guest is Professor George Perry. He's a professor of neuroscience, developmental biology, regenerative biology at the University of Texas, San Antonio. He's also former dean of the College of Sciences there. He's very well known for his work in oxidative stress and brain aging and Alzheimer's disease. He's the editor of the Journal of Alzheimer's Disease, has been for a long time. So he has a broad understanding, I think, of basic biology, and we'll talk about that. Actually, his training was not in clinical realms or anything really obviously to do with Alzheimer's disease, and it's kind of, as I talk with him, you'll see how just a basic understanding of evolution, biology, how cells work, has implications for humans and both in health and disease. So George, welcome.
Thank you very much, Mark. Quite an honor to participate with you.
It's going to be fun. I haven't seen you for a long time, and I ended up moving out to well, you were fairly close physically, actually, for a while. I was in Kentucky, University of Kentucky, and you were in Ohio, the bordering state at Cleveland Clinic, and that's where we first crossed. But you have an interesting background. So you're from California originally, and
Correct.
And I don't know if you lived on the coast or not, but somehow you got interested in marine biology, I guess. Can you talk about that?
I grew up on the coastal California, near Point Conception, which is a very isolated, continually isolated place. And it marks the place where North and South California occur, the currents change. So I had quite an appreciation for the biology of the region, including the marine biology, which was generally untouched and still is because it's now a national reserve.
So you spent time on the water then a lot?
Not so much in the water, more in the, at the intertidal region.
I see.
My father was really into this, the water thing. He would go fishing all the time. I wasn't that excited about fishing.
And you mentioned, so is the climate change affecting the ecosystem there yet or not?
I don't know. I was just there, not at Point Conception, but I was just in California. I don't know if it really is affecting things that much, but it's certainly at that point, on one side of Point Conception, you see one type of groups of animals. On the other side, you see another group of animals.
Huh, that's interesting.
Because the current.
So you went to UC Santa Barbara?
Correct. I went to UC Santa Barbara and I grew up very close to there. I grew up 40 miles from UC Santa Barbara.
And both you and I are undergraduate degrees in zoology. Actually, I wanted to go into veterinary medicine, but I wasn't smart enough.
I doubt that, Mark.
But my younger brother, I wasn't actually that good at just memorizing things and spitting them back. It wasn't. I got kind of bored with that kind of almost monotony type thing. But my younger brother was better, and he is a practicing veterinarian. Actually, he's teaching in a veterinary college in West Virginia.
Oh, great.
And then he went to a really neat place, but San Diego, I guess Palo Alto region, Scripps, right?
I did my PhD at 3 locations. The degree is from Scripps Institution of Oceanography, part of UCST, but much older than UCST. But I spent half the time at Stanford's Marine Station in Pacific Grove. called Hopkins Marine Station. And I spent 3 summers at Woods Hole.
Oh yeah, I've been one summer I taught part of one of their summer courses. It's a great place. And did you so what was your PhD dissertation? What was the kind of the main take-home message of what you
The main take-home message of my PhD is actually not far from what I study in Alzheimer's disease. I studied calcium control of oxidation, non-mitochondrial oxidation, lipid peroxidation, and naphthoquinone oxidation, both that were calcium controlled.
Wow, I didn't even know. So we had overlap there too, because I did a lot of work on, calcium and it's
I knew that, as we're used to. So
and so there's
my
calcium.
science family is considered to be the one that initiated the idea of calcium control in cells. Daniel Mazia, well, my director was David Epple, but his mentor was Daniel Mazia, and his mentor was Halbrun from the University of Pennsylvania, who is, I think, the first person who suggested calcium was important and was not regarded as being correct. And that was during the 20s and 30s.
Yeah, and it's it's certainly in many ways the most important ion in nerve cells. So go ahead and talk about reactive oxygen species. You mentioned mitochondria, which is a location in cells most people think of as a source of, but just go back to like oxidative stress 101. What is oxidative?
You know what? I'm not certain I progressed very much beyond oxidative stress 101. I've been studying it since, wow, the mid 70s and 75. I think it's right. Yeah, 1975 summers when I started. And I still don't understand it, which is why it still interests me. If I understood it, then I'd probably be bored. So what I studied in sea urchins is fertilization. And during fertilization, it was well known from the early 1900s that there's a big change in oxygen consumption in some eggs. In sea urchin eggs, they increase oxygen consumption at least 5-fold within a few seconds after fertilization. And some of that is cyanide insensitive, which means that it's likely not mitochondrial.
Because the cyanide inhibits the electron transport chain.
Yeah, it stops it at the very last point because it substitutes for oxygen. And that was why I was looking at what were the properties. And if homogeneous of eggs, you could add calcium and they would consume oxygen.
And calcium is an important signal when the sperm and the egg.
Within 15 seconds. First, there's a depolarization. And then about 15 seconds later, with fertilization, you can time things down to the second, if not better. And that calcium wave is associated with the oxygen increase. And so anyway, one of the things that's important to note is that fertilization is pretty essential to the survival of the egg. If it doesn't get fertilized, it just degenerates. It's where if it fertilizes and goes through this stress and all those things, it's going to become an embryo. So why does that start? Well, from the basis of that, oxidative stress is not necessarily a bad thing, okay? Yeah. So back to Alzheimer's disease. By the time I obtained tenure, and I was studying the cytoskeleton, and I think we met during that period, is that I decided what was I going to do something that I really enjoyed doing, which was close to marine biology. And that was to study oxidative stress in Alzheimer's disease. And I invested, I think, 7 years to produce the first paper, to try to figure out some simple method to demonstrate there was something that was related to radicals beside lipofuscin. That doesn't mean I don't think lipofuscin is important, but I'll tell you, that's way more complicated even than I understand right now. So we were looking at these changes, thinking that the most important thing about them would be that plaques and tangles, the lesions of Alzheimer's disease, have very long residence times. So they were bound to accumulate some type of oxidative changes, and that might underlie their insolubility in detergents and other chaotropic agents. Well, we found that there was glycation, which is sugar-related modification, and lipid peroxide modifications. But as we started looking at these more carefully, we found something that was quite a surprise. And if I had been careful and thinking about what I was observing, I would have noticed it right in the beginning. But it became more obvious when I looked at oxidative modifications that were not secondary. Lipid peroxidation and glycation is you have an oxidative abnormality, produces some intermediate, reacts with something else, and it's more of a chain of events. In the case of nitration, or in the case of oxidative carbonyls, or in the case of nucleic acid oxidation, that's a direct oxidative event. It means it's produced right at the location in which you see it. And when we saw that, we saw something that really was, for me, quite surprising. And that is that oxidative damage involves every neuron of a certain category and involves their cytoplasm. And it had very little to do with whether there was a pathology there or not. If there was a tangle within the neuron, actually they had less oxidative damage, 50% less. And if there were plaques in the area, there was also less, whether you looked at amyloid within the cell or amyloid outside. So that led to a couple ideas. One, which I'll go to later, is the idea that maybe many of the pathologies are protective responses and part of the biology of aging. Instead of the cause of the disease, they're associated with the cause. And that's why they correlate so well. The other one was, what's abnormal in a neuron that looks normal? Because there were not many descriptions. I mean, in retrospect, Ralph Nixon had described lysosomal abnormalities. At about the same time we were doing these experiments. But so we decided to look what was within the neuron that was abnormal, and we focused on mitochondria. Because there had been prior work, Flint Beal, John Bloss, and others, that suggested there were some mitochondrial abnormalities. And we decided to do it very much like we did for oxidative stress. And what we did in oxidative stress that really made a big difference was to make all of the approaches in situ type approaches. If you just grind up the tissue, you really don't see much of a change. In fact, I did a meta-analysis with Dr. Schrag recently. And in fact, if you analyze them, there's not much of a change. And that's what we found. We started by doing bulk analysis and found very small changes, statistically borderline. So therefore, I never reported any of them. I shouldn't say. I reported 8-hydroxyguanosine, but I reported it in a chapter because the findings are really small. And so when we used antibodies, and in the case of looking for mitochondria, we did it by in situ hybridization, using either light level work or EM work with gold nanoparticles. And we found that there was a big increase in mitochondrial DNA. We did a morphometric study of biopsy tissue, tissue obtained from living people with Alzheimer's disease, and found mitochondria, if anything, are reduced. And in the EM result, we found that all this extra mitochondrial DNA was in autophagosomes.
Yeah.
A tremendous amount of it. I mean, of course, it was in mitochondria, but most of it was in autophagosomes. And I haven't reported this. It happens when people are in their late 30s.
It's interesting. So I was at National Institute on Aging for a long time, trying to understand. So everything starts to decline in the 30s, I think. Certainly does from sports, performance, physical performance and athletics. for pretty much any sport you can think of, your performance starts to decline. So, aging process is starting earlier than most people would like to believe, or maybe
Yeah.
that you and I would like to believe. But so the mitochondria, yeah. So I had David Rubenstein on as a guest talking about autophagy and mitophagy, which is when mitochondria get dysfunctional, and apparently often when they're not functioning well, they produce more superoxide, more free radicals. And then, so you're saying you discovered that they're accumulating in these autophagosomes, and how does that relate to the amount of oxidative damage in the specific neurons in which these are accumulating?
Great question. Actually, I have not published all of the analysis, even though it's been done for a long time. If you look at it in normal aging,
Well, we're not going to scoop you, George.
no, I'm not worried in
I don't
anything.
I don't have a lab anymore. I'm retired.
Okay. Is that if you look at this from normal aging, the type of changes that occur when people are in their late 30s all the way out in age, there's a strong correlation. It's actually 0.86.
Ah,
If you quantitate, densitometry of how much mitochondrial DNA is in a neuron as a population, and you compare it with how much oxidative damage that neuron has. These are lower levels than you find in Alzheimer's disease, but they're extremely correlated. When people have Alzheimer's disease, it's higher. Both are higher, but they're uncorrelated. That's the paper I still haven't put together. None of us in my collaborating group have ever figured out the understanding, because there's a transition between Alzheimer controls from normal aging to Alzheimer's disease is not a continuum regarding what we studied.
okay, so we have to consider also that in Alzheimer's disease, some neurons are dead, and some probably neurofibrillary tangles that are there are not representing living cells, right?
We quantitate each cell singly.
Okay, but do you know whether it's alive or dead?
Well, it has to be a cell.
And this is in postmortem tissue?
Yeah.
Okay, so it has to have a membrane and
Yeah, it
not,
has to have a cell with
not just a
cytoplasm.
tangle.
We're
Not
not looking at extracellular tangles.
Okay.
Actually, all the neurons, aside from those that have tangles, skipping extracellular tangles, but intracellular tangles, whenever they have tau accumulations in the cell, oxidative stress is reduced by 50%.
Interesting.
And then if we look at the quantitation of this, once people have Alzheimer's disease, the correlation then is related to amyloid, and it's inverse, always inverse. In many conditions, sporadic Alzheimer's disease, Down syndrome,
And this is
and every condition we've looked at, genetically caused Alzheimer's disease, PS1 mutations, all of them have the same property.
now, my lab, some in collaboration with Bill and David Butterfield, but a lot not. So we have this evidence that this lipid peroxidation 4-hydroxynonenal, which covalently modifies proteins on one of the 3 different amino acids, cysteine,
Yes.
lysine, histidine, was increased in association with the pathology of Alzheimer's. And then when we either directly expose cultured neurons to this 4-hydroxynonenal lipid peroxidation product itself, or we expose them to amyloid beta peptide when it's in a vigorously aggregating state, we found that certain membrane proteins are covalently modified by this aldehyde. And the ones we found were pretty interesting. The sodium pump, the glucose transporter protein. So that suggests right there that it's going to cause problems with the cells controlling their excitability and in the cells getting enough energy. And so we kind of developed this story that, that there's oxidative stress related to aging and maybe some disease-related processes, and that's leading to this membrane lipid peroxidation, and that's screwing up the ability of the neurons to maintain proper excitability. And there's evidence that patients with Alzheimer's have increased incidence of epilepsy, and there's other evidence there's hyperexcitability early on. There's certainly impaired glucose transport early on. So we're thinking that this particular aspect of oxidative stress may play an important role. Have you looked at, and then I think that, yeah, we had one paper that tau, this protein that's aggregating and it's a main part of the neurofibrillary tangles, can be modified by this aldehyde.
Yes.
So maybe it's sopping up the aldehyde and preventing it. If you're thinking it's protective, maybe it's. You see what
Yeah, no, we've done experiments along the same line.
Yeah, yeah.
In the case of tau, we found that it's very susceptible to carbonyl attack, whether it's hydroxynonenal or glucose-related ones, and it's controlled by phosphorylation. The tau is not modifiable unless it has phosphates on it.
Yeah.
And when we analyzed antibodies that were raised to tangles, to neurofibrillary tangles, everyone I've analyzed has an oxidation aspect to the epitope.
Oh, wow, that's interesting.
And they're different for different antibodies. So the structure that people describe where it comes together, there appears to be some sort of cross-linking piece. And then the most dominant protein we found that gets modified is a protein that has a lot of sequence similarity to tau, and that's neurofilament heavy and medium subunit. And has the same property again. If you get recombinant neurofilament, which has no phosphates on it, you can't modify it. Or if you take neurofilaments and strip off the phosphates with phosphatase or hydrofluoric acid, you also can't modify it. So it seems to be needed having exposure.
It's an attempt of the cell to protect itself against.
That's what I thought, but I'll tell you, I'm still super confused because in the case of neurofilaments, the modification, I studied it in rodents and humans and rabbits, and I found essentially the same results in all. The one I published was mice because of the comparison with transgenics, is that we found that the neurofilaments were modified as soon as they were phosphorylated. And it did not increase either with age, and I received mice or bought them from the NIA repository, to get the maximum age without having to age them myself. So I think I studied 39-month-old mice, the oldest I could buy from them, and, increments all the way back to 1 month. But I'd studied also, not in the paper, embryos also, but I didn't include them. And I found that the modification was always the same.
Yeah, so that's interesting, the embryos. So the tau, as you mentioned, is hyperphosphorylated in Alzheimer's and the tangles, but it's also heavily phosphorylated during that kind of vigorous stage of brain development when neurons are growing and forming synaptic connections.
Yeah. Correct. And so the other piece I got from this, number one, is that oxidative stress is far more complicated. And that I think one of the issues is that a lot of these modifications are signaling pathways that we don't understand very well. Or I thought initially, and I still think, is that it could be exactly what you're saying, is playing the role as a scavenger. But if it was playing that role, then you would expect to see accumulation over time. And also in the case of the mice, I inhibited proteolysis with calpain inhibitors. I didn't see any change for anything I did. Okay. It just became modified.
Was
The
this
case of the
particular,
tangle,
George,
pardon
was
me?
that particular modification with 4-hydroxynonenal? So I mentioned the cysteine residues, right? And there's this intrinsic antioxidant, it's a tripeptide, glutathione, right?
Yes.
So it has a cysteine in it and two other amino acids. And there is evidence that glutathione detoxifies 4-hydroxynonenal, and there's even evidence that the cells spit it out. So what if, so this will be good for the listeners because we're just kind of brainstorming. What if, is there evidence that cells spit out tau?
Oh, with the exosomes and other
Yeah,
things
and
like that, a
maybe,
lot of it
so maybe it's
suggests that.
continuously being removed, kind of like, so you don't see its accumulation in the cell.
Correct. But in neurofilaments, I don't know, because theoretically it's all in polymer. Theoretically.
Yeah.
And that's very theoretical because I was just really surprised. So I tried looking for reversal mechanisms
Uh-huh.
with Larry Sayre and a student I had
Oh.
with peptides to see if we could find, but the modifications were very broad based because some of the modifications are reversible. But we found the same result for modifications of H&E, like the fluorescent product, which is a cross-link. There's no indication that it's ever reversible. But, it doesn't mean it isn't possible.
Yeah, it's covalent, so
There'd have to be some kind of enzyme or
yeah.
something. And none has been described, although I have seen some work on glycation that suggests that there are mechanisms to reverse it. You asked about the tangle and the reduced thing, the reduced oxidative stress. Well, the reason we can explain it, and once they explain it quantitatively, explain it mechanistically, is wherever we see tau accumulations in neurons, we see an accumulation of heme oxygenase.
Huh.
And that's an antioxidant enzyme. It's an antioxidant enzyme that takes heme, which is a pro-oxidant, and converts it to an antioxidant, biliverdin and N-bilirubin. But why is that important? Well, if you have mitochondrial turnover, you're liberating lots of hemes, and you need to metabolize them. That isn't said when we made the discovery of heme oxygenase. We weren't thinking about mitochondria, but they always occur together. And if you change heme oxygenase expression, you change tau phosphorylation. They are very correlated, So and then metals are involved in a lot of these things. The other thing that mitochondria have is they're very rich in iron and copper.
Yeah.
Bill Marksbury did some of the fundamental studies in this area with another Mark Level. And I don't know if you were involved in those studies. I don't remember.
I was there when they were being
That's what I
done.
thought.
So they use that interesting technique. They essentially take a brain section and it's called laser activated microprobe elemental analysis. Anyway,
Lemma.
they're essentially like doing, what do you call it? They kind of burn a hole. What it looked like to me when they showed it, they're like burning a hole in the cell and then determining what metals, so they did, all sorts of the aluminum thing. They kind of hopefully put that to rest, the idea that Alzheimer's caused by people eating out of aluminum pans. There's not much aluminum in the tangles. And they did, there is iron. And did he do copper? I can't remember.
Yes, he did.
He did copper.
He did copper also, at least in one of the studies.
Yeah. Yeah,
so
go ahead.
Well, so the broad range of oxidative damage we found usually is catalyzed by metal-related things. So when you mention mitochondria produce reactive oxygen species, most of those reactive oxygen species are not that reactive. they need to actually be catalyzed to be able to perform something like a hydroxyl radical, which then oxidizes the molecule it's attached to. I mean, because it's so reactive that it hits there. So therefore, we looked in those same neurons, were there reactive metals? Not just metals, but metals able to do oxidation. And then Mark Smith, my close collaborator, developed some methods to look at redox activity within neurons and lesions, and found that the plaques and tangles had lots of redox active metals. We thought that they were iron mostly, and that may be still the case, but I will tell you more. We refined that answer significantly. And then, but also the cytoplasm had this, and it happens early. During mild cognitive impairment, you see an increase in reactive iron within cell bodies.
George, that so normally we have iron binding proteins, right? All sorts of them, ferritin and so on. And so iron is normally bound to proteins and it doesn't damage us. It's when it gets in the ionic Fe2 plus form when it can catalyze these reactions you're talking about.
Correct. Most of the time, the body tries to keep the metals, especially copper, but also iron, attached to proteins which control its redox activity. And that's going to underlie what I think is happening in Alzheimer's disease. So we found that there was more redox activity. And if you looked at a young person, like 20s, and that's where we started to change also our idea of a control. And age-matched control was useful, but we also needed young controls. And young meant under 40, not 50 or 60.
Well, and another control that people often don't use is a disease control. In other words, like Parkinson's or like, something else, some other disease, so that, is what you're seeing specific, say, to Alzheimer's or not?
We did the mitochondrial studies, this in situ studies in Parkinson's, and the results are quite different. Superficially, they seem like they're the same because you have an age-dependent change. But when you start doing it more carefully, you find that that's all there is. There's an age-dependent change, and Parkinson's don't accumulate mitochondria and autophagosomes in the same way. It doesn't mean that there isn't an autophagosome problem, but it doesn't lead to the DNA. So the DNA accumulation in autophagosomes is not restricted to just that. We found lipoic acid, which is a prosthetic group for mitochondria. Enzymes also in autophagosomes, in that case, even in lipofuscin. And cytochrome oxidase, immunoreactivity, was not in the autophagosomes. It was loose in the cytoplasm. And that's kind of mystifying because, the current ideas of apoptosis, if you release mitochondrial contents of the cytoplasm, the cell should die. Well, that's happening when people in their late 30s, at least some people, and by their 50s and 60s, everybody. And why aren't these neurons dead? And then, of course, if they have Alzheimer's disease, they again persist and there's massive release. So we looked for metals. First was just developing some methods to look for redox activity rather than the total metals. We also did some assays for total metals and didn't find anything really that spectacular, so it's not reported. And then we tried refining that over and over again. For one of the things is Mark Smith found that the plaques and tangles were magnetic. In other words, they'd move to a magnet. And it took me, 2 decades to figure out how to describe what we were seeing. And what it is that there are small magnetite crystals that are super paramagnetic. They're actually not magnetic in a sense. They're magnetic when they're in a magnetic field. So we've described that now carefully with standard physical techniques as opposed to us with a magnet in our hand moving something. And so we found this magnetite. We further analyzed, and the magnetite we had an atomic resolution microscope, a TEM that has resolution of 0.6 angstroms, and we could see the actual crystal structure. And then we recently worked with Joanne Collingwood in England, collaborative work. We've used X-ray microscopy with the synchrotron source in Oxford and Berkeley, and been able to get at exactly what is the redox state. I mean, if you find magnetite, you know it's iron 2, iron 3 couples. But when we looked at this for copper, we didn't know how it is. There are not really great methods for looking at copper and looking at its redox state because the specificity is not great. But in this particular case, we were able to determine iron 2, iron 3, copper 1, copper 2. So those are all redox pairs. But the thing that was the most surprising, and to our knowledge has never been documented in any animal before, the dominant form was metallic, both iron and copper. There was abundant. And this is with isolated plaques. We've now completed a study with plaques in situ, in which there's been no treatment, because, it could have been created from the treatment, the isolation procedure. And the same results are been obtained. So what do we think's happening? Well, number one, I don't know how this stuff formed. Okay. And even more perplexing, how it's stable. Because we've also done this similar study, but it's not ready for publication yet, of tangles. And in tangles case, the preliminary work suggests that there is not these metallic forms. There is accumulation of iron and copper, but they're not in metallic form. And in earlier work, we showed that amyloid beta stops lipid peroxidation, copper-induced lipid peroxidation. But I think that's only part of the story.
So iron, metal can rust, right? It's oxidized.
Yes, that's why I think it's crazy that there could be metallic, because we don't put any special care in storing these. They're stored in a refrigerator or freezer with atmospheric air.
You'd have to do it under nitrogen or figure out a way to.
But iron stays even in the presence of oxygen.
Yeah, that's right. Okay.
That's why I say that.
Yeah. So
what I think is that the plaque, either the amyloid or some other component within the plaques. We've also the isolated plaques, when you do mass spectrometry of them, you see that amyloid beta is not the only component by any means. In fact, it may not even be the dominant component. And that's probably also true for tangles. We haven't done that, is that these things are really complicated structures. So all of this points to us that there's a lot more going on. And we think that oxidative stress doesn't mean that the disease is caused by oxidative stress, but oxidative stress is an important element that's dealt with, because you can't ignore radicals and you have to get them into a physiological situation, or else the cell would die immediately. Of course, we know they eventually die, but they don't die immediately.
We mentioned, we haven't talked about synapses much, and I think it'd be worth spending a little time on that, because you got synapses where let's talk about a glutamatergic synapse. By the way, I wrote a book, Sculptor and Destroyer.
Wow. I was seeing it there, and
yeah, I've written.
No, I can't see it's you if I get close enough. Okay.
But anyway, it's all about glutamate, which is in the lay public. It's interesting if I ask most people, even psychiatrists, interestingly, what's the brain's most important neurotransmitter, they may say dopamine or serotonin, but more than 90% of the neurons are glutamatergic, and all the work most of the work on learning and memory has to do with what's happening at glutamatergic synapses, and certainly if there's any role for excitotoxicity and Alzheimer's, and a lot of the neurons that die in Alzheimer's are glutamatergic. But anyway, so the synapse get glutamate's released, activates postsynaptic receptors, which are ion channels, either sodium or calcium, so the AMPA or NMDA receptor. So you get sodium influx, calcium influx, a burst of reactive oxygen species, right? And there are mitochondria. These synapses are often way away from cell bodies. I think you're studying cell bodies mainly, right?
Correct. But I'll tell you some work about synapses.
Okay, go ahead.
Well, in terms of oxidative modifications, synapses do not show so much change, okay? But we just completed a study a couple months ago. It was actually done because of COVID. It was analyzing micrographs I left at Case Western Reserve University, and Jean-Wei Zhu and Sandra Sidlak analyzed these micrographs, as well as collecting new ones, biopsy tissue, looking at the synapse and what was altered in synapses in Alzheimer's disease, with a focus on mitochondria. Because prior studies they had completed, is on the cell body, the proximal portion of the axon hillock, but to trace the whole axonal environment was beyond our skill level. Maybe that's possible now, because of the ion beam process, but to do it otherwise serial section through it, it's not realistic. So in any case, so they went and looked at what happens at synapses. And what they found was major problems in mitochondria, much like we found in the cell body, and that the synapse had vesicle problems. Not so much, the paper doesn't emphasize this, there wasn't so much a loss in synapses. What there was a disruption of synapses, where the vesicles were away from the active zone. There's reduced number, and the reduced number was correlated with how many mitochondria were there.
Huh. That's really interesting. Yeah, you've got to have energy. It's important to appreciate. There's got to be a lot of energy, ATP, available at synapses, because that's where a lot of the ion fluxes occur.
Correct. It's a highly energetic area. Yeah. And if you have disruption of that energetics, the issue of finding oxidative damage there, I've never noticed it in synapses, which also quite mystified me. Okay. Axons, yes, they have this carbonyl modification on neurofilaments, principally on neurofilaments. But in synapses, it was not striking. Yeah. But there's certainly a striking disruption of this. And this result actually is quite consistent with prior work. Another one of your colleagues in Kentucky, who I view as really the father of the whole synapse business, Steve Schaff.
Yeah.
I greatly admire his work. So we were extending his work. He found that there were synaptic changes, but they weren't huge. They were more of synaptic remodeling and things like this. In fact, I called him not so long ago just to make certain I wasn't confused about what he found.
He's retired now, I think.
Finally. Yes, that's what he told me. I tried to get him involved in a company I was in also, and he said he wasn't interested in doing science in any case.
But he had a student, Pat Sullivan. Have you ever come across Pat?
No.
Oh, he's done quite a bit of work on mitochondria, and something we can talk about in a little bit maybe is the idea of how can we bolster the mechanisms by which neurons deal with free oxygen radicals and damage and so on. But so we did all this work on intermittent fasting, some on exercise, and then also intellectual challenges, most of it in animal models, and had this that's consistent with epidemiological data, right? That people's brains keep working better longer if they're in good general health, if they exercise, if they keep their brain exercised, and if they don't overeat, take in too many calories. And in fact, with the calorie restriction and the intermittent fasting, those are only two ways, at least in rats and mice, that you can reliably and markedly increase their lifespan. So I guess we can talk then, we're talking about synapses and mitochondria. We've done work suggesting that exercise and maybe intermittent fasting stimulate over repeated cycles, over days and weeks and months, an increase in the number of mitochondria in individual neurons. And we did some work where in experimental models where we can prevent mitochondrial biogenesis by what we did, we used RNA interference to suppress levels of a protein called PGC1 alpha. And we initially did it in cultured neurons as they're developing and forming synapses, and found if we prevent mitochondrial biogenesis, it impairs the ability of synapses to form between neurons. And then in the adult brain, we actually knocked down PGC1 alpha in hippocampal. We did dentate granule neurons. We had a viral vector, knocked it down. And anyway, there, over a period of several weeks, there was a loss of synapses if the neurons can't keep, presumably producing more mitochondria. So there's got to be like this constant turnover. I don't know what the time course is, but turnover of mitochondria. And if that doesn't occur, that actually can cause big problems at synapses. Yeah.
But I also, by finding this and it's consistent with Bob Terry's work and Moslius, that they talk, because they used vesicle stains and defined those as synapses, right? Synaptophysin.
Yeah.
And for me, synaptophysin is not equivalent to a synapse. It's equivalent to a piece of a synapse. So our findings actually are consistent with them. There is a synaptic vesicle change, but the synapse change is different than just loss. And it probably means, I think, that, restoration is more possible. That if you knew what to do, things are there. It isn't that everything is gone. So one of the ideas, and then we've done a lot of studies on mitochondrial dynamics, mainly fusion-fission. Yeah. And that fusion-fission properties is systemic. That isn't just neurons. Even though the neurons show these oxidative abnormalities and the other cells don't show it to the same extent. If we look at fibroblasts in culture, they show almost the same changes.
And so this is fibroblasts from patients with sporadic Alzheimer's? Or
yes.
Familiar?
Yeah, the ones from, the NIA repository.
Yeah, yeah.
The ones anybody can, buy from the system.
Yeah.
And the mitochondria in Alzheimer's disease are larger in people with it. Not, they're not giant mitochondria, which people describe. They're just larger. So when you look at the micrographs, you may not even notice it. But they're nearly twice as large. And that's also true for fibroblasts, even though you culture them. And it's paralleled with changes in protein expression.
And this is compared to age-matched controls.
Age-matched controls. And if you change expression level of one of the, we've changed them for a number of proteins, but I mainly focus on one. Dynamic like one protein, you change the size, you can change its expression.
That's very important. So so fibroblasts, I think, my impression, we could rule out any role for amyloid accumulation or tau. The fibroblasts don't even express tau.
Yeah.
Right?
And yet they have, I don't think so. I mean, there are other cells that do express them, but I don't think so.
No.
So they have the same mitochondrial change, not to the same extent. You know, the neurons have massive
And
amount of oxidative.
did you do fibroblasts from young people too?
No,
No.
only age-matched people. We did it for oxidative markers initially, and then Jean Zhu did it for looking at mitochondrial dynamics
So
and did quite extensive studies in it. And there's alteration, different proteins. Some control energy production, some control radicals. They're all altered. There's an Alzheimer type pattern, and there's a control type pattern. The neurons have it more extremely, not surprising, and fibroblasts have it much less. So the other part, so that's at the dynamic protein, the fusion fission proteins, and others that control mitochondria. And then the other part is that there's a change in microtubules, a massive change, having nothing to do with tangles, at least statistically. Since I had thousands of micrographs of biopsies, we counted the number of microtubules in those micrographs, and either the length or the number, we did both. And we found that there was about an 80% reduction in people with Alzheimer's disease versus age-matched controls. In that case, we did look at the aging phenomena, even though we have a really small number of cases, and it was extremely correlated. From age 60 to 80, there was a drop in microtubule density among the normals, but the Alzheimer patients had way lower microtubule density, and whether there was a tangle there or not had no statistical difference.
You know, George, so this again started when I was at Kentucky. It was
I remember you doing microtubule studies.
well, we did a lot with calcium, right? And because
Well, calcium could be the overall.
that's what
When
I'm
you're
thinking.
looking at such pleiotropic changes, calcium is certainly a real suspect for doing it, right?
Yeah.
It changes mitochondrial because that's one of the main things that they pump is calcium. So it disturbs mitochondrial. It disrupts microtubules.
There was a colleague of mine, and I'm blanking on his name at the time. So we were using these new calcium indicator dyes that Roger Chen developed, so you can image relative calcium levels. FARA2 was the first one. And then there was this other guy at Kentucky, and Siskin. His name was Siskin.
I don't know him.
He was using a quaron, which is a fluorescent.
Yes, from
But he published
jellyfish.
papers, and this is in the 90s. He published papers where he looked at calcium regulation in fibroblasts from Alzheimer's patients compared to age-matched controls and found alterations in calcium. Actually, it seemed like they were accumulating higher levels of calcium than they normally would.
And that could be, you know, if we address that, answer is no. Okay. But if you were really looking at something that it has such a broad effect, calcium could be the driver. But here, as it is in sea urchins, as it is in a lot of systems, calcium controls oxidative phenomena, and lipid peroxidation controls phospholipase C, you know, which eventually generates free arachidonic acid or other unsaturated lipids.
Main source of 4-hydroxynonenal?
Yes, arachidonic or some others like it. And there are, it is not restricted to only 4-arachidonic acid. I mean, 4-hydroxynonenal is, I think, Alan, you also looked at acrolein. There's a whole gamish of carbonyls that are either produced from direct oxidation on proteins or things, or secondary products.
Yeah, and John Carney was involved in that a little too.
Correct. You guys were some of the first people to be doing that.
Yeah.
Carney, Bill Marksbury were really fundamental.
Yeah, I miss Bill a lot. The only reason I seriously considered staying at Kentucky, even though I was getting like a wonderful offer from the National Institute on Aging in terms of my setup and the number of positions I have and so on, was Bill. You know, he was a great guy, great mentor. Good scientist, good clinician. So I miss him a lot. Let's talk about, so people who are listening to this who aren't really up on like maybe the cellular things going on at the molecular level, they want to know, so what's the implications of this for people in terms of, can they reduce their risk for Alzheimer's, or what's on the horizon for clinical trials? There's been universal failures of this amyloid, trying to remove amyloid approach. And there really haven't, the only drug that has any ability to slow the progression, and it's like, it's not much at all in the clinical trials, is actually a glutamate receptor blocker called memantine, which it's not a strong effect. The problem with glutamate is it's so important. It's essential for our learning and memory. It's essential for everything the brain's doing, that if you start messing with glutamate signaling, that's going to cause a lot of problems. And this memantine drug, it's very sensitive. You know, if you increase the dose a little bit, it causes side effects, even psychosis, kind of like ketamine. And actually, my father had, I don't know if you may not know the story of my father, because
No, I don't.
this was after. Okay, so we grew up on a farm. My father was actually the prosecuting attorney for Olmsted County, Minnesota. I went to Mayo High School. And a lot of the kids there in my classes were doctors' kids, you know, Mayo Clinic. And we trained and raced harness horses.
Well, that I knew.
Yeah, standard, but here's some bits from a couple of our best horses. Okay, so my dad retired. He was only 60 years old when he retired from his county attorney position. And then my mom, unfortunately, she was a heavy smoker. She was overweight. Anyway, she died from a severe stroke at age 67. So then my father was alone. The kids me and my brother and sister had moved away. So he was alone on the farm, you know, beginning from the age of like 67. And then when he got in his early 80s, when I talked to him on the phone, so I'm in Maryland, he's back in Minnesota, he would start asking me the same question more than once during a conversation. It could even be like a 10, 15-minute conversation. And for people who are listening to this, that's a sign that there's maybe something going wrong with short-term memory. When a person doesn't remember, they've already asked you a question. Hopefully I haven't asked you more than the same question twice.
I've been writing them down.
And anyway, so we took him into the Mayo Clinic to, guess who? Ron Peterson.
Yeah, that's what I was going to say.
Yeah, Ron Peterson. He was Ronald Reagan's doctor. He's done a lot of nice work on mild cognitive impairment, and they have a nice Alzheimer's research center there. And so he diagnosed my dad with probable Alzheimer's disease based on the cognitive test. And then we enrolled my dad in their Alzheimer's research center program. You have until 11, you have another 20 minutes?
Yes.
Okay.
Yeah. No,
I'm doing this at home.
No, I have this down exactly for that time.
Okay. And right, so every year he'd go in, they'd repeat the battery of cognitive tests. They would do MRI, looking at brain imaging, and they actually didn't take cerebral spinal fluid from my father. He didn't want to do that. My brother and sister, you know, we had to. But anyway, over the years, it progressively decreased. Actually, the one thing that was consistently decreased from year to year, even more than the cognitive testing, which can kind of fluctuate, although over multiple years it's going down, was the size of the hippocampus. This brain region that's important for learning and memory. It did progressively decrease. And then finally, when my father was just 90 years old, he passed away. My brother went and lived with him for 5 years, which was nice. And then the last year or so, we put him in a kind of a facility that specialized in people with dementia. But I think part of his cognitive decline might have been because after my mom died, he no longer had any or much at all social interactions. He didn't read a lot after he retired from being a lawyer. He never read a lot. I'm just, guessing. But also he was old. But the reason I mention that is they did autopsy, and Dennis Dixon actually did the pathology on my dad's brain. He didn't have Alzheimer's. He had what they finally said was mixed dementia. He had some amyloid, but it wasn't marked. He did have neurofibrillary tangles, but not. What he did have is massive loss of neurons in the hippocampus, something that Peter Nelson, who's at Kentucky.
Yes, who's really the pioneer in that area.
Yeah, he calls it hippocampal sclerosis of aging. Now, the reason I point this out is that I mentioned my dad was diagnosed with probable Alzheimer's disease, and the criteria for Alzheimer's disease is it's a kind of arbitrary, based on some neuropathologists getting together and saying, okay, if there's this density of amyloid plaques and this density of neurofibrillary tangles, if it's that or above, we'll call that Alzheimer's disease. If it's below, we won't. But I think, this idea that Alzheimer's disease is like something completely different than any other dementia it's not true. What are your thoughts on that?
Oh, I completely agree with what you've said.
Yeah.
It's a collection of changes that people call Alzheimer's disease. And also, as you pointed out, describing your father, is that the disease of the aged, those people in their 80s continuing to the 90s, is usually different than those people that have it when they're 60. Almost back to the pre-senile dementia, which was only Alzheimer's, and only changed during the 1970s. I think that's problematic. I know I had that discussion group that we on the web of people, and a paper that we just got accepted, and they said, well, we want to change the definition. And I said, I don't think we really want to go down that path, because it's really hard to know. I mean, if you look at it historically, as time has gone on, Lewy body disease would have been classified as Alzheimer's disease. Frontal temporal dementia would have been classified. It depends on how careful you are in doing the analysis.
Yeah.
So by having brain banks and collecting a large number of people like, the careful pathologists like Peter Nelson and others are able to define these subgroups that are more homogeneous. I think there's going to be more of them as time goes on. Will they be truly homogeneous? Because there's a change of normal aging.
Yeah.
Even if you're completely normal, you're going to end up with plaques and tangles in your brain.
Yeah.
The other part, so that's one part. The second part is back to what I mentioned before. There's all this stuff, why the drugs have failed, because too late, because the brain is all destroyed. That's variable feature also of the disease, how much neuron loss.
Yeah.
From studies from the early 80s of Bob Terry, who's a careful anatomist, he found that there was no correlation. Where he found the correlation was the synaptophysin with cognitive loss.
Yeah.
And if you look at people that are older, not all of them have, like your father, massive cell loss and atrophy. Some have a really minor amount of atrophy. Yeah. There isn't a good correlation of it. Yeah. It's correlated, but not one-to-one.
So all these disorders, aging is a major risk factor, the disorders we talked about. And so in theory, if you get an intervention that fundamentally impacts the normal aging process, that should be beneficial in all of these. And if you look at this, like step back and say you compare to the cancer field where, have different cancers and it could be solid tumors, it could be, white blood cell cancer, whatever. They are trying to develop, specific treatments for individual types of cancer. But on the other hand, the treatments that can be effective are kind of generic treatments that, in the case of cancer, the idea is you want to kill the cancer cells with causing as little possible damage to your normal cells. In the case of Alzheimer's, frontotemporal lobe dementia, Parkinson's, we want to do the opposite. We want to keep the cells alive in all of those disorders.
Alive and turn them to being functional.
And functional, yeah.
Because I think what's happened in Alzheimer's disease for these synaptic changes is that the neurons are there. Eventually they die, but they're there initially, and they're not functioning correctly. Something fundamentally changes, whether it's being driven by calcium or it's being driven by metabolic failure or all of the above. It's how do you bring them back to functionality? Or one of the ideas we had was that the neurons have undergone some type of reaction like hibernation, in which that they've moved their metabolism to a protective response, but a non-functional response.
The one clinical trial, so when I was finishing up at NIA, we got interested in, as I mentioned earlier, if you do PET imaging and look at radiolabeled glucose uptake,
yes,
in the cells in the brain, it's impaired very early on, as early as MCI patients and so on. And then this goes way back to our cell culture experiments, which I mentioned, where we found that oxidative stress, iron, actually, we found ferrous iron, A beta, at least when it's in a certain state, and when it's association with the membrane, can impair glucose transport. So then the question is there another energy source you can give to neurons? And the most obvious one is ketones. And there are specific transporters in the membrane of neurons called monocarboxylic acid transporters, MCTs. And we think the MCTs are not impaired. The cells, Steve Quinane, do you know his work? Up at Montreal. So he did these really beautiful studies where he did PET imaging. First, he did it in just normal kind of middle-aged adults. He simultaneously injected into the blood radiolabeled glucose, 2-deoxyglucose, and so 2-deoxyglucose radiolabeled, and then radiolabeled acetoacetate, which is one of the two ketones that our cells can use for energy. These ketones come from our fatty acids in our fats. So he imaged in the same people simultaneously relative levels of glucose utilization and ketone utilization. And then he had the subjects go on a ketogenic diet. So essentially no carbohydrate intake for, I think he did a week, maybe 2 weeks. Then he redid it. When the person's in the ketogenic state, the cells are using mainly ketones. And now there's at least 2 clinical trials going on of this ketone ester beta hydroxybutyrate in AD patients. One trial is by a neurologist who was under me at NIA, Dimitros Kapagiannis, and the other is Steve Quinane's group up in Canada. So this is, I'm semi-excited about this. The history of clinical trials is fail, fail, fail. But we'll see what happens with that.
No, it's a very interesting idea about change in metabolism and change in the balance from there. How could one change calcium balance without disturbing things? without being worse than where you started?
Maybe using your neurons.
Well, the only thing the only thing that really changes the course of Alzheimer's disease after you have it, maybe, but certainly before you have it, are these type of changes: exercise, diet, stress reduction, being socially involved, as well as other things, the much broader if you're deficient in something. But all of those, and we can see that when you compare the difference with minorities. Those are mostly, we don't know completely, but it's likely that most of it's being driven by socioeconomic issues.
Yeah.
And those socioeconomic issues are differences in exercise, difference in diet, blah, blah, blah. And that means you have double. So you're not going to be able to escape Alzheimer's disease, but you're going to be able to increase your health span.
George, I went on CDC. I got interested in kind of the demographics of various diseases, and they have these nice maps where, what's the prevalence of obesity, what's the prevalence of type 2 diabetes, and a lot of these, it's on a county-wide basis throughout the country. And so you go on there, and there's it's the Southeast. So Mississippi, Louisiana, Texas somewhat.
Yeah, Texas is one of those.
But what they have now is they now have some data on neurodegenerative disorders, also, dementia, Alzheimer's. And what's happening is there has been an relative increase in prevalence. And this going back like 30, 40, 50 decades ago, increased prevalence in those areas. So my interpretation is kids are growing up in families that have poor health habits. They're taking too many calories. A lot of it's junk food, processed food. They're not getting much exercise. And then these kids, and there's data that, so I think if we can get these kids in shape, and they'll do better academically, and because a lot of these areas, the kids don't do so well in terms of academic achievement, going to college, and getting a good job. And so we've got this transgenerational issue that I think is going to compound bad things going on in the brain as our population is aging.
I agree with you completely that the public health aspects of this are really critical. And if you were looking at that, if you're looking at heart disease or some aspects of cancer, you'd see similarities.
Yes.
That your risk in the aging process about how you age. It doesn't mean that people are going to point, well, this person did all those things and they still got Alzheimer's. Okay, but they probably developed it 10 years later than they would have otherwise.
But you can say that with smoking too, and nevertheless, the government finally cracked the whip on the smoking thing. But it's a bigger issue in regulating, unhealthy foods and stuff, or sugar and fructose, high fructose drink. that's a big problem too. And how do you? Yeah. Well, that's a whole.
Anyway, but there's a lot more information about this. I've reviewed, I don't know, in the last couple of years, like 4 or 5 books on this topic. So I think it's actually penetrating to some degree. And those disparities, between white America versus African American versus Hispanic, I think people have to dwell on the fact that it really relates to socioeconomic issues of those groups.
Yes.
And not probably
not genetics. Yeah, it's not genetics.
Correct. It's unlikely to be genetics.
No.
And the genetics certainly plays a role, like some groups have more ApoE, but the penetrance even there depends on diet, as you probably know. It's really common among Africans, but Africans ApoE4 doesn't make a huge difference in their risk because of their diet, probably because of their diet.
Yeah, that's a good point. And also, the only genetic difference I know for sure, I'm by far an expert on this, is risk for skin cancer.
Oh, yes.
It's higher, and you and I have much higher risk for getting skin cancer than do African Americans, and simply because they have better genes in that respect, and the melanin, right, in the skin protects the skin.
Yeah, we're adapted to our environment, and they were adapted to an environment that had more sun, and you were adapted to an environment that had less sun.
Yeah. Okay.
Anyway, that's where, but I think that needs to be one of the things, and I'm glad you and I both agree that the current treatments, the antibody treatments, really are not effective. They're very borderline, and they subject patients to a lot of risk.
Yeah, and I think you were one of the first ones to point out a long time ago, even when they were getting started on some of these trials, that this rationale may not be good. I even put in a postdoc put in a small note to Science on this. We messed around with different A-beta antibodies in culture and found that some of them actually potentiated the toxicity of A-beta. And then tau, I think the same thing's going to happen with tau. There's this efforts to, I think, right, to target some treatments for tau. I think we got to target more energy metabolism, something more generic, something more fundamental to the.
Yeah, I think this comes up that the people who are driving these views have a really poor biological understanding of evolution. Things that happen commonly in well-adapted organisms, not organisms that have mutations, because that's a defect, but amyloid, tau are normal things that everybody has and even have during development, as you were talking about. We just completed a study. In fact, Craig Atwood was the leader of that, one of my former trainees who's now in Wisconsin, showing that tau phosphorylation was critical in neuronal differentiation. So
it makes sense because you have it controls the polymerization state of the microtubules, for goodness sake. And the microtubules are like, my postdoc work, I think you may know this, I was studying regulation of growth cones, dendritic growth cones, hippocampal neurons, and I was finding glutamate plays a role in, but for the axons and dendrites to grow, you've got to get polymerization of the microtubules, and then they're changing depending on kind of the plasticity that's going on.
And calcium is the major thing. That's what I did for my postdoc studies.
And by the way, mitochondria move along microtubules. A lot of things actually move along microtubules, so they're like railroad tracks that are moving things around in a coordinated manner. Yeah, so you have a protein tau that affects their polymerization. It's a kind of influence all these things. Yeah. Okay, George. Boy, I could talk with you. This has been really fun. I could talk with you the rest of the day, I think, because. But one thing I did want to mention. There are ways to bolster the antioxidant defenses of cells. You mentioned heme oxygenase. There's a lot of interest in actually food supplement things and these chemicals in plants that seem to be good for health. And some of them upregulate our antioxidant defenses. There's this pathway called the NRF2 ARE pathway that upregulates the heme oxygenase. But there's other pathways. And one way to actually upregulate antioxidant defenses is to use your neurons. just like muscle cells, when you exercise, there's a huge increase in free radical production in your muscle cells when they're exercising. But it's been shown pretty convincingly that if you don't get that increase in oxidative stress, there's this nice study by this one group who they loaded up people with vitamin E and vitamin C, and then they exercised them and compared to people who weren't loaded up. And actually, that impaired the training effect. So these reactive oxygen species have critical signaling roles. And one thing they do actually is upregulate our intrinsic antioxidant. Yeah.
We've done, well, who did that type of experiments was Paula Moreda, one of my trainees who's in Portugal, Coimbra, Portugal. And one of her students came to our lab and completed them and looking at hypoxia adjustment or glucose. And glucose can be toxic in some conditions. And one of the things that protected it was mild hypoxia, or in cells, mild cyanide.
Yeah.
And if you put an antioxidant in, there was no protection from the hypoxia.
Yeah. This
So
is
it was about
preconditioning or hormesis or whatever you want to call it.
correct. And then the daughter of my PhD mentor, Alyssa Eppel, has done a lot of studies in humans with stress and low levels of stress, and how that's at many levels stress, psychological stress, as well as physical stress. We're talking about low levels, not PTSD
I'll have to look
levels.
her up. I'm working on a third book now. I wrote one on the science of intermittent fasting, then the glutamate one, but the third one is on adaptive stress responses, hormesis.
She wrote a very nice review in what is it, gerontology, something or other. Her last name's Epel. She's at UCSF.
Okay. I'll look that up.
She's a major thought leader in that area.
Yeah. I may have seen her papers and just, yeah.
And she uses a term I can't remember. I tried to get her to write a review for JAD about this issue.
Well, but Bruce McEwen used the term eustress. Eustress. And then a lot of people use this hormesis thing like
and people have changed even the term of oxidative stress. Now people change the other one to oxidative distress. Because there's two types. There's the type when you're thinking of chronic stress, it's sustained and it actually induces protection to have a new balance. As where you have acute stress, like say you got head trauma and you were going to die, or a stroke, and things become out of equilibrium and everything dies consequently. And those two things, and a lot of the description of oxidative stress really relates to the distress idea.
Yes.
Rather than the homeostatic one, which is balanced, but it's not the same as it originally was.
Yeah.
So you're not the same when you're 80 as when you're 20. No. You may be close on the outside, but on the inside, you're not functioning the same.
Oh, I know. I used to run a lot, and then I started to have some knee problems. So I switched to mountain bike. And then I had an accident 4 years ago. And so what happened, the way I interpret it is that, when I was a kid, I used to race motocross, race dirt bikes. But the bottom line is, I'm pretty sure the reason I crashed is because my reaction time, I'm in my 60s, right? And I'm still riding on these really hard technical trails with steep hills and rocks and everything. And and I had to have 3 surgeries after this.
Okay.
And so we think our brain, we think we can still do things we can do when we're young, but we actually can't do them, or can't do them as well anyway, even though we think we can. Yeah.
Correct. But so I think that when you're looking at this whole issue, like blueberries, in fact, Jim Joseph used to really get upset with me because I would say, what's the difference between blueberries and cyanide? And meaning that, plants produce so many toxins that are so critical for their protection.
Yes.
And we don't eat the ones that kill us.
Right.
We eat the other ones. And the other ones still have toxins, but
Yeah.
they're toxins we tolerate. And the same thing is exercise. If you over-exercise, you can cause damage to the system too.
Yeah.
But if you cause small amounts of stress, the recovery of that has value. I mean, whether it's taking a class that tests your brain or learn, training to run a marathon or
Yeah,
5K or
it's
whatever
got
it
to
is.
be stress recovery cycles, stress, then recovery period, whether it's resting after exercise
See,
or, sleeping or,
I can't hear you now, Mark.
oh, really?
Oh, you're back in.
I was
Oh,
just saying,
my connection was
also,
unstable.
also important is the cyclicity. You stress, good stress, exercise, fasting, eating potentially toxic phytochemicals, then a recovery period. And actually, the phytochemical thing is interesting because we've evolved at least 4 ways to deal with these toxic chemicals. One is taste. All these chemicals that are good for your health have a bitter taste. The second is vomiting. The third is cytochrome P450s in the liver that rapidly detoxify them. Then the fourth, which we come to the hormesis, is cells respond adaptively to these by, for example, upregulating antioxidant defenses. So we've evolved all these systems because there's other things in these plants. They do have energy, right, which is good for us. So it was to our advantage to be able to tolerate these so that we can get the benefits.
Yeah, and the simplistic view that the plants have benefit just because they have a lot of vitamin C and vitamin E is way too simplistic. Of course, they have that, but that isn't the main benefit because all of the data says that the benefit is from eating plants, varied types of plants.
Yeah.
And those varied types of plants have way more. Otherwise, vitamin E and vitamin C and et al. Would have a tremendous health benefit, which they don't unless you're deficient.
Yeah.
And that's when I, talk to family members about they want to know if somebody is demented. It is useful to know whether people have nutrient lack because
Yeah.
people can be B12 deficient or other things.
Yeah.
But the idea of eating lots of spinach and blueberries or any other phytochemical rich fruit and vegetable really has a major impact, and minimizing meat and other things.
Yeah,
like
I agree. We're on the same wavelength. And by the way, you look very good, George.
Oh, gee.
For me not having seen you for so long.
I'm 70 this year.
I know you don't look 70. Okay.
Well, thank you.
Anyway, we'll finish up the podcast now. Again, thanks for taking the time. I know you're busy, and I enjoyed seeing you again and kind of brainstorming and talking about these things. And have happy holidays.
The same to you. It's really been a pleasure, Mark.
Okay.
Quite a pleasure.
Bye.
Hope we have a chance to meet personally again shortly.
Yeah, me too.
Thank
Bye.
you.